Hello everyone. In this video, I will tell you about DNA fingerprinting. So let's get started. DNA fingerprinting. As you all know, DNA is the master molecule of all life form. I think you know the full form of DNA, deoxyribonucleic acid. Okay, and this DNA, it constitute the blueprint of every living organism through which characters of an individual are passed generation to generation. Okay. Every individual, every living organism differ only due to nucleotide sequence of chromosome. And that nucleotide, form code. Therefore, this coded genetic information can be profiled to produce most authentic identity of an organism. Means every individual differ only in nucleotide sequence of chromosome, and that nucleotide form the code, and therefore this coded genetic information can be profiled to produce the most authentic identity of an organism. The identification of an individual at genetic level is known as DNA fingerprinting, or more specifically, it is DNA profiling. Okay, this is the just introduction. Now, who developed this technique? In 1984, Al-Zafrey developed the technique of DNA fingerprinting. I'm writing short form of fingerprinting. Okay. Now, from the Human Genome Project, we know that only 99.9% base pair sequence is the same in all. Okay. That means only the short stretch of the sequence which is responsible for giving the unique identity to an individual, it's only a 0.1% base pair sequence which is differ person to person. This information we know from the Human Genome Project that 99.9% base pair sequence is same in all individual in all the living organism. Only 0.19% base pair sequence is differ for person to person. And this DNA fingerprinting is specific for every individual, and a person will have same DNA fingerprinting in a cell, tissue, organ. And this DNA fingerprinting cannot be changed. It cannot be changed by any method. Like our hand fingerprinting, it will be changed by some method, some techniques. But this DNA fingerprinting, at any cost, it will not change. Now, we centrifuge our DNA sample in centrifugation tube. For that, we are using in it cesium chloride density gradient. Okay, let's take, this is the tube of centrifugation. In this cesium chloride is there. In this tube, we are getting the two bands. The one is at the end, bottom of the tube. This is the broad band, and we are getting another band at the top of the test tube. This one is narrow band. In a broad band, density is increased and it is rich in GC. In narrow band, density is decreased and it is rich in AT. A for adenine, T for thymine, G for guanine, C for cytosine. Okay. This narrow band, it will represent repetitive sequence or repetitive DNA. Okay. Now, this repetitive DNA or repetitive sequence is further divided into two types. Let's take this is the chromosome. Here any sequence is present. That is, I am drawing here. DNA is a double stranded structure, but here I am drawing only the single structure, a single strand for simplicity. Let's take a sequence like A, T, T, G, A. A, T, T, G, A. A, T, T, G, A, T. This is the sequence is present here. What we are seeing here, this sequence is repeating at the same site. The sequence which is present here is repeating at the same site. This is known as tandem repeat. Okay, the next type is like A, T, T, G, A. This sequence is present. Here also we are seeing the sequences are repeating, but at different side, and this is known as intersuppressed repeat. Okay. Now here in the first one, this repeats is getting how many base pair? Like 1, 2, 3, 4, 5 and 6. 6 base pair. On the 6 base pair, on the basis of this, the standard repeat is again classified into the two type: short base pair or long. In short, 60 base pair is there. In long, more than 60 base pair is present. And this short base pair, which is having the 60 base pair, is known as satellite DNA. Okay. Again, this satellite DNA is further divided into two types. In this, in first type, 11 to 60 base pair are present. In second type, 1 to 6 base pair is present. 11 to 60 base pairs are present. This is known as mini satellite. And the second one, in which 1 to 6 base pair is present, is known as microsatellite. This mini satellite is also known as VNTR, which is variable number tandem repeat. Microsatellite is also known as STR. It stands for simple tandem repeat. For DNA fingerprinting, we are using this VNTR, and this VNTR gives the unique sequence that will be utilized for DNA fingerprinting. Okay. Now we will see the method of DNA fingerprinting. For this, first we have to isolate the DNA. Isolation of DNA. First, cells are broken down to release DNA. If only small amount of DNA is available, it can be amplified by using PCR. PCR is polymerase chain reaction. This is the sample. Okay. In this we have only a small amount of DNA. Now by using PCR we can amplify it like we get the amplified DNA. Now the second step, from this we are getting this our DNA. Okay. Second step, digestion of DNA. The DNA which we have is cut into fragment by using restriction enzyme. Here we are using endonuclease restriction enzyme. This endonuclease restriction enzyme cut the DNA at a specific base sequence. This is our DNA. With the help of restriction enzyme, it will cut into the fragments. Okay. Now it will yield thousands of DNA fragment which is of different size. Now the third step is separation of DNA fragment in gel electrophoresis. DNA fragments are separated on the basis of size, for that what we have to do is, this is our gel electrophoresis. These are the wells. We have to add our sample into it. Here agarose gel is present. This is positive and this is negative. Now DNA fragment are injected into well. In this we have to inject our DNA fragment and then electric current is applied along the gel. We know that DNA is negatively charged, so it is attracted to the positive end of the gel. Shorter DNA fragment means those DNA fragment which are in short will move faster than longer fragment. This is our gel electrophoresis. These are already injected with the DNA fragment. Now DNA is negatively charged, so it moved to the positive end. The shorter fragments move faster and reach to the positive end. After this will be hybridization with radiolabel VNTR. A radiolabel material is added which combine with DNA fragment and produce a fluorescent image. Now this will be read as autoradiography. The pattern of fragment which we are getting is distributed and it is then analyzed. This is will get these type of pattern, and we have to then analyze this. Once again, I'll summarize this. First, we have to take our cell, that cell with, first we have to collect the sample. That sample which contain the DNA. If the DNA sample which is we are taking is present in a very small amount, that will be amplified with the help of PCR, that is polymerase chain reaction. After that, the DNA is cut into the small fragment with the help of restriction enzyme, particularly endonucleases. After this, the DNA cut, this restriction enzyme cut the DNA at the specific base sequences, and this will yield the thousands of DNA fragments which is of different sizes. After this, we have to separate the DNA fragment in gel electrophoresis. This is a gel electrophoresis apparatus. These are the wells, and in this agarose gel is present. Now we have to inject our DNA fragment into this wells, into this weld, and apply the electric field along with the agarose gel. Now, as we know that DNA is a negatively charged, it is attract to the positive and it start moving. The shorter DNA will move the faster, and longer DNA will move slowly. After this, we have to mix the radio label VNTR with the DNA fragment, and that will produce the fluorescent image, and that will image which was the image which is found, we have to analyze that pattern. This is the method. Now, what is the application where it is used? I'll tell you the broad application of DNA fingerprinting. The 5 broad application I will going to tell you is, the first one is in forensic science. The second one in parenting testing. The third one in medical diagnosis. The fourth one in food industry. And the fifth one is in agriculture. DNA fingerprinting is used to solve crime and medical problem. Let's take an example. DNA fingerprinting can solve crime. How it can solve the crime? Let's take a crime is happened in some area. There, forensic science team get a sample of a blood. They get the sample of blood and they did the DNA fingerprinting. They analyze. After the analyzing, if the pattern of the first of all, they get the pattern of a DNA profile, which is then compared with those with those of the victim and the suspect. If the profile matches the suspect, it will provide the strong evidence that suspect was present at crime scene. The matches of the profile with the suspect, it does not mean that the person did that crime. It just gives the evidence that the person was present at the crime scene. If the profile does not matches, then the suspect may be eliminated from the inquiry. Okay, let's see. They get the certain pattern. This is for suspect. They get for blood sample. They get from the crime scene, and it is from victim. How they get the strand pattern? This is the pattern they got. From this we can say that suspect and blood sample are the same and the victim has a different pattern of DNA fingerprinting. And this DNA fingerprinting also solve the problem in medical